Very good morning to all. So today we have to move to the one more cell organelles, that is the endoplasmic reticulum. We already completed the ribosomes, mitochondria, plasma membrane. Next we have to move, that is the endoplasmic reticulum. So this slide indicates the introduction of the ER. ER is nothing but the endoplasmic reticulum. So in the year 1945, the lace-like membrane of the endoplasmic reticulum were first seen in the cytoplasm of the chick embryo cells. Means where first time these observed from the chick embryo cells, that is in the year 1945. It looks like a lace, like a membrane. Okay, these are membrane bound channels seen in the form of a network, delicate strand and vesicles in the cytoplasm. How it will be looks like means it is a very delicate and network like structure arised from the nucleus, okay, membrane of the nucleus. These are single membrane cell organelles. Outermost it consists of only one membrane, that is the single membrane cell organelles. These form an interconnected network of tubules, vesicles and cisternae within in cells. Means what parts present in the ER means tubules, vesicles and the cisternae present within the endoplasmic reticulum. ER considered as one of the component of cytoskeleton, along with the microtubule, microfilament and intermediate filament. Means all these are the cytoskeleton part, that is the microtubule, microfilament. Along with that, ER also called as the one of the cytoskeleton in the cytoplasm, means which gives the which hold the cells and which gives the perfect shape and it will be maintains the cell properly. That is the main function of the cytoskeleton. These are first of all observed by these scientists, that is the Porter and Fullman as a network like structure. The term ER was first used by the Porter and Fullman. These two scientists used the term ER. Where these are located, where the endoplasmic reticulum present in the cell, present in almost all eukaryotic cell, absent in the prokaryotic cells. These are found to be absent in mature erythrocytes, embryonic cells and prokaryotes. Means all the eukaryotic cell contains the ER, but prokaryotic and the erythrocytes, these will doesn't contains the ER. The ER often occupies most of the cytoplasm. Where the ER present, mostly it is occupied in the cytoplasm of the cell. How many number of the ER present? That is the amount. The ER varies in amount from cell to cell. In spermatocyte, it is represented by few vacuoles only. Okay, each and every cell have the different number of the endoplasmic reticulum. Spermatocytes have the few vacuoles only. In the cells of adipose tissue, it is quite simple, having the form of a few tubules. Some have the vacuole-like structure, some have the tubules. Only few number of the ER will present in different cells. The cells that are actively synthesizing proteins such as liver and pancreatic cells and fibroblasts have abundant ER. Okay, means all this tissue or all these cells have the more number of the ER, because if the cell secreting large number of the proteins, then ribosomes also in large number and the ER also in a large number. Okay, the very active cell have the large number of the cell organelles. Endoplasmic reticulum forms 30 to 60% of the total membrane in cell. This much number of the ER present in a cell. Origin of endoplasmic reticulum. From where it is originated? At present, manner of origin of the ER is not definitely known. It is not conformly known. The most concrete hypothesis is that ER is budded off from the nuclear envelope. Most of the scientists will think that ER is oftenly originated from the membrane of the nucleus, that is the nuclear envelope. Outer membrane of the nucleus we call as the nuclear envelope. From that side only it is originated or it is developed. The ER appears to arise from the outer membrane of the nuclear envelope by outfolding or from the plasma membrane by infolding. How it is originated? It may be from outfolding from the nucleus, that is the nuclear envelope, or infolding from the plasma membrane. The smooth ER seem to arise from the rough ER by a detachment of ribosomes. Actually, in the ER, two types are there: smooth endoplasmic reticulum, rough endoplasmic reticulum. Smooth endoplasmic reticulum is originated from the rough endoplasmic reticulum by detachment of the ribosomes. Attachment of the ribosomes takes place one by one. When they are separated, immediately the ER will be originated. This is the structure of the ER, endoplasmic reticulum. So see the structure properly. That is the smooth endoplasmic reticulum, rough endoplasmic reticulum, ribosomes, cisternae, nuclear pore, nuclear envelope. All these parts comes under the endoplasmic reticulum. So, if the ribosomes are present on the surface of the endoplasmic reticulum, then we call it as the rough endoplasmic reticulum. If the membrane of the ER doesn't contains the ribosomes, then it is the smooth endoplasmic reticulum. So, the ER structure is like this. There are two basic morphological types ER, that is the rough endoplasmic and smooth endoplasmic reticulum. The ER membrane is thinner 50 angstrom than that of plasma membrane. It is too much thin as compared to the plasma membrane. Okay. Then next, physical structure of the ER, endoplasmic reticulum. Physical structure is nothing but how it will be looks up externally, morphologically. The ER is three-dimensional network of intracellular, looks like a three-dimensional structure. It is formed of three types of the elements: cisternae, tubules and vesicles. Mainly these three components present in the endoplasmic reticulum. First we shall study the cisternae. These are flattened unbranched sac-like element. Okay. So cisternae, see the structure here. This part we called as the cisternae, which is flat, unbranched and the sac-like element. The line stacks parallel to one another. They are formed parallel to each other. They bear ribosomes on the surface that therefore appears rough. So cisternae contains the ribosomes on its surface. That's why it is the rough. That's why the name given as rough endoplasmic reticulum. It contains glycoproteins named ribofourin 1 and ribofourin 2 that bind the ribosomes. So the nature of the cistern is nothing but the glycoprotein. In nature, the name of the glycoproteins ribofourin 1 and 2. So these will be glycoprotein helps for the binding of the ribosomes on its surface. Next is the second one, tubules. These are irregular. Okay. Means tube-like structure present in the ER. These are irregular branching elements which form a network along with other element. These are often free of ribosomes. Okay, these doesn't possess the ribosomes. Vesicles, next part in the ER, vesicles. These are oval and rounded vacuole-like element. These will be looks like a vacuole-like structure. Okay. So vesicle is referred as the round structure in the ER. These are also free of ribosome. Only cisternae contains the ribosome. Vesicles, tubules doesn't possess the ribosomes. All the elements of ER freely communicate with one another and contain a fluid called endoplasmic matrix in the ER lumen. So all the elements means vesicle, tubule, cisternae, all these are communicate with each other and form the matrix like or the fluid like part that called as the endoplasmic matrix. These matrix is different from cytoplasmic matrix outside the ER. ER matrix is different and the cytoplasm is different. Means cytoplasm is also fluid, that is also different, and the fluid present in the ER, that is also different. The ER may pass from one cell to another through the plasmodesmata in the form of desmotubules. So these are present in the cytoplasm. So these will be passed from one cell to the next cell. So this is the structure of the ER. Okay, this is the nucleus, nuclear pore, nuclear envelope. From the nuclear envelope, ER is originated. Smooth endoplasmic reticulum, rough endoplasmic reticulum. Ribosomes present on the surface of the ER. Then cisternae space, cisternae, vesicles, all these are located inside. Then next is the molecular structure. What chemical composition is present in the ER, that we have to study. The membrane of ER are composed of two layers of phospholipid, phosphorus plus lipid, present in the membranes of the endoplasmic reticulum, sandwiched by two layers of proteins molecule like other membrane in the cell wall. So types, that is the smooth endoplasmic reticulum, rough endoplasmic reticulum. Endoplasmic matrix means it is the fluid part present in the ER. The space inside the tubules and vesicles is filled with the watery medium that is different from the fluid in the cytosol outside the ER. That only I told, the matrix or the fluid of the cytoplasm is different and the fluid of the ER is different. Their walls are constructed of lipid bilayers, made up of the lipid bilayers membrane that contains large amount of proteins similar to the cell membrane. How the plasma membrane consists the chemicals, like that only it is also consist. Smooth ER. So what do you know regarding the smooth ER? Smooth ER is an arrangement of tubules, vesicles and the sacs. Here ribosomes are completely absent. The size and structure of the SER varies between the cells. Actually 2 marks question going to be asked, expand SER, RER. Okay. So you have to write smooth endoplasmic reticulum and rough endoplasmic reticulum. The size and the structure of SER varies between the cells. The SER can change within the cell's lifetime to allow the cell to adapt to change in its function and requirements. So it will be changed within the cell. SER can be changed within the cell and it will be adapted for the functions. There are no ribosomes attached to the membrane surface. Here ribosomes are absent. The SER is connected to the nuclear envelope. So the directly SER is connected to the nuclear envelope. Then RER, rough endoplasmic reticulum. The surface of the RER is studded with ribosomes, giving it a rough appearance. That's why the name given as rough endoplasmic reticulum. It mainly consists of cisternae. The membrane of the RER forms large double membrane sheets which is located near and continuous with the outer layer of the nuclear envelope. Okay, the membrane of the RER, it forms the double membrane sheet, so that located near and continuous with the outer layer of the nuclear envelope. RER is very important in the synthesis and packaging of the proteins. Actually, most of the protein synthesis will occur in the rough endoplasmic reticulum by the ribosomes. Okay, that's why ribosomes located on the ER, that is the rough endoplasmic, the synthesis of proteins will be takes place. Binding site of the ribosomes on the RER is the translocon. Means it is actually related with the protein synthesis. Then how the protein transport occur? As proteins are formed in the endoplasmic reticulum, they are transported through the tubules towards proteins of the SER. Means the protein secreted by the rough endoplasmic reticulum, and that will be transported to the tubules towards proteins of the SER that lie nearest to Golgi apparatus. Means each and every cell organelles are connected with each other. When the ribosomes are present on the ER, that secretes the protein, that will transfer to the tubules, then it is transferred to the smooth endoplasmic reticulum, then to the Golgi apparatus. At this point, small transport vesicles composed of small envelopes of smooth ER continually break away and diffuse to the deepest layer of Golgi apparatus. It will be go to the deepest layer of the Golgi apparatus or Golgi bodies. Inside this vesicle are the synthesized proteins and other products from the ER present. How the transportation will be occurs? Like this. Okay, rough endoplasmic reticulum at work. Firstly, nucleus is there, then rough endoplasmic reticulum, then vesicles, Golgi, then cell membrane. Like this, movement of the proteins will be takes place up to the cell membrane, then it will be passed to the other cell also. Functions of RER, SER and the sarcoplasmic reticulum, that is the SR. What is the function of RER? Surface for ribosomes. Okay, the RER provides the space for the attachment of the ribophorins for the attachment of the ribosomes. Surface for protein synthesis. Actually, the main protein synthesis occurs in the RER. Formation of glycoprotein. Linking of sugars to 4 glycoprotein starts in the rear and it is completed in the Golgi complex. Okay, so sugars will be combined to form the glycoproteins that occurs in the rear. Synthesis of precursors. The rear produce the enzymes precursors for the formation of lysosome by Golgi complex. Even one more cell organelle is there, that is the lysosome that arises from the Golgi complex. So, RER produce the enzyme that will be helpful for the synthesis of lysosomes from the Golgi apparatus. Smooth ER formation. The RER gives rise to the smooth ER by loss of ribosomes. When the ribosome will be moved away from the ER, then the smooth endoplasmic reticulum will be formed. Functions of SER. The smooth endoplasmic reticulum lacks ribosomes and functions lipid metabolism, carbohydrate metabolism, detoxification, and is specially abundant in mammalian liver and gonad cells. Okay, so the breaking down of all the food takes place with the help of SER. It also synthesizes phospholipids. Okay, cells which secrete these products, such as those in testes, ovaries, and skin oil glands, have great deal of smooth endoplasmic reticulum. Most of the synthesis of the phospholipids occurs in all these cells. Detoxification will be occurs with the help of SER. Brings about detoxification in the liver, that the drug poison into harmless ones of excretory by the cells. Formation of organelles takes place with the help of SER. Lysosome, Golgi apparatus, vacuoles. Then it also carries out the attachment of receptors on the cell membrane, proteins and steroid metabolism. So attachment of the receptors will be takes place on the cell membrane, proteins and the steroid metabolism. In muscle cells, it regulates calcium ion concentration. Means in muscles actually calcium is present, so that metabolism or regulation will be takes place with the help of SER. The smooth endoplasmic reticulum also contain the enzyme glucose 6-phosphatase, convertase glu-6-phosphatase to glucose, a step in gluconeogenesis. Okay, glyconeogenesis, all these process requires the enzymes. Those enzyme secretion takes place with the help of SER. SR, that we called as the sarcoplasmic reticulum. So, sarcoplasmic reticulum is smooth ER, found in smooth and striated muscles. Actually, SR we called as the sarcoplasmic reticulum present in the muscles. The only structural difference between this organelle and the smooth endoplasmic reticulum is the midlay of proteins they have, both bound to their membranes and drifting within the confines of their lumens. This fundamental difference is indicative of their functions. Means in their difference in the proteins and membranes, that we called as the SR. The endoplasmic reticulum synthesizes molecules, while the sarcoplasmic reticulum stores and pumps calcium ions. Means it is present in the muscles, that's why the regulation of the concentration of the calcium ions takes place with the help of SR in the muscle cells. The sarcoplasmic reticulum contains large stores of calcium, which it sequesters and then release when the muscle cell is stimulated. So the maintenance of the Ca or calcium takes place with the help of SR, because it is the storehouse of the calcium. So when it requires the muscle, on that time synthesis takes place. When it will be released by the muscles, that will be occurs by the presence of SR. It plays a major role in excitation, contraction, coupling in the muscle cells. Okay, so nerve contraction will be takes place, that is with the help of these SR. Okay, so up to this regarding the endoplasmic reticulum. So if you have any doubts, you ask, or you can message in group. Okay, so next time we can study the lysosome. Okay, thank you.